All the King's Men, Enteric Glia. The enteric nervous system, which I will call ENS, in Greek enteron means gut. This system is part of the autonomic nervous system that regulates the functioning of the gastrointestinal tract. The nerve plexuses that make up the ENS are located in the walls of the hollow organs of the gastrointestinal tract. Some neurophysiologists suggest that similar plexuses are located in the hollow organs of respiratory and genitourinary systems and unite them under the term metasympathetic nervous system. Enteric neurons and glia develop from a general population of neural crest cells migrating to these organs. The ENS of the gastrointestinal tract consists of three plexuses: the subserosal, myenteric, and submucosal. Major populations of enteric gliocytes are found in enteric ganglia in the myenteric and submucosal plexuses. These cells are also found within the muscular layer and in the lamina propria of the mucosa. The subserosal plexus is located between the serosa and the muscular layer of the intestine and has a few small ganglia. In the autonomous ganglia of the ENS, neurons and gliocytes are densely packed, being closely connected by nerve bundles containing axons of both internal enteric and external sympathetic, parasympathetic, and primary sensory neurons. The ganglia, in turn, are interconnected, forming networks or plexuses that extend along the entire length of the digestive tract. Unlike other autonomous ganglia, connective tissue and blood vessels are not found in the ENS ganglia under normal conditions. General morphology, outer structure, and relationships with bodies and processes of neurons of enteric gliocytes are very similar to those of astrocytes. Like astrocytes, enteric gliocytes are stellate cells with a large number of highly branched processes. Most cell bodies are filled with nuclei. Like astrocytes, enteric gliocytes often receive synaptic contacts from neurons, which indicates that these contacts are the main morphological substrate of neuron-glial relationships. Like astrocytes, enteric gliocytes express glial fibrillary acidic protein and S100 with antibodies to which they can be detected. All of the above allowed some authors to call enteric gliocytes astrocytes of PNS. However, even though these two types of cells have much in common, there are certain differences. They indicate that enteric gliocytes are fundamentally different from astrocytes. Firstly, enteric gliocytes develop from the neural crest, while astrocytes develop from the neural tube. Secondly, enteric gliocytes do not express an astrocytic marker for aldehyde dehydrogenase. Thus, when speaking about the astrocytic properties of enteric glia, it is necessary to remember its unique nature. A feature of enteric glia is that enteric glia populations are located in different parts, myenteric and submucosal ganglia, within the interganglionic neural pathways, directly beneath the mucosal epithelial cells, and are associated with nerve fibers located between smooth myocytes. And although all enteric glia sites come from a common pool of precursors, the neural crest cells, and express a common set of biomarkers, the phenotype of mature enteric glia sites depends on the microenvironment in which they are located. Consequently, interactions between enteric glia and other cells of the intestinal wall lead to glial diversity in the ENS. Attempts have been made to classify various types of enteric glia sites based on morphological differences. Unique classification for glial populations based on morphological differences between the enteric glia was proposed. While some enteric glia sites and protoplasmic astrocytes are similar due to the presence of short, irregularly branched processes, these glia sites were named protoplasmic or type 1 enteric glia sites. The elongated glia sites within interganglionic fiber tracts resemble fibrous astrocytes, and they were called fibrous or type 2 enteric glia sites. Later, this original morphological classification was extended. Subepithelial glia with several long branch processes could be classed as mucosal or type 3 enteric glia sites, and the elongated glia running with nerve fibers in the musculature as intramuscular or type 4 enteric glia sites. However, unfortunately, the generally accepted classification of enteric glia sites has not been developed yet. This is a consequence of differences among the obtained experimental data because each enteric glia population located in different parts of the wall of a hollow organ is a functionally different subtype of enteric glia sites. The ratio between neurons and enteric glia sites in different animals ranges from 1 to 4 to 1 to 10, in humans 1 to 7, which indicates the important role of gliocytes. Functions of enteric glia: maintaining homeostasis in the enteric ganglia. Enteric glia sites in enteric ganglia participate in homeostatic mechanisms supporting neurotransmission of neurons. Enteric glia sites supply neurons with the necessary precursors for the synthesis of neurotransmitters, including nitric oxide, glutamate, and gamma-aminobutyric acid. Enteric glia sites also support neurotransmission by regulating the bioavailability of neuroactive substances in the extracellular environment. Enzymes on the cell surface of the enteric glia sites and various transporters remove neuroactive compounds surrounding enteric neurons. Similarly, channels in the glial cell membrane support neurotransmission and prevent excitotoxic neuronal death by regulating and buffering extracellular potassium. Regulation of neuro and glia transmission. In addition to the homeostatic mechanisms discussed above, enteric glia sites are actively involved in the neurotransmission of neurons and are able to detect and modulate their activity. Enteric glia sites have a set of receptors which allows them to monitor the activity of neurons and initiate intracellular signaling mechanisms in response to the release of neurotransmitters. Enteric glia sites modulate enteric neurotransmission, releasing neuroactive substances, which is called glia transmission. In particular, it was shown that an increase in the concentration of calcium ions in the intestinal wall initiates the release of ATP, which is a neurotransmitter and neuromodulator in the ENS. Barrier functions. The intestinal barrier limits the penetration of bacteria, viruses, and toxic substances into the body. Intestinal subepithelial enteric gliosides are in close contact with epithelial cells, nerve fibers, and blood vessels. This glioside population is a key regulator of intestinal barrier function. Enteric glia in tissue culture release several factors, transforming glial growth factor beta-1 and proepidermal growth factor, which affect differentiation, adhesion, migration, and proliferation of epithelial cells in monolayer. Evidence of in vivo enteric gliosides barrier function is based primarily on modeling ablation of these cells. It was shown that the permeability of the epithelium increases after the destruction of enteric glia. In models on transgenic mice, where enteric gliosides were selectively removed, the integrity of the epithelial barrier was impaired and inflammation occurred in the intestinal wall. Thank you for attention.